Thank you for the introduction, and thank you for the invite, and it's wonderful to be able to speak in front of you all today and with such an important topic. And so, I'm going to sort of briefly introduce you to some of the work that my group is doing in the Department of Chemistry here at King's, and particularly we're focusing on how we can, as it was nice to introduce, how we can utilize biology and harness it for the things that we can need for our modern life, but hopefully in a more sustainable manner. And that really is the problem. So our modern way of living is basically generating a lot of stuff. We need to produce a series of materials that have only really existed in the past 50 or so years. Those are plastics, as you might imagine. There are lots of other materials as well that we need to use. Lots of chemicals. Even medicines require high energy sort of requirements to synthesize and to produce, and on the volume that we require. And this comes with two key problems that we have sort of moving forward if we want to protect not just how we live, but the planet we live on. The first of those is that energy requirement. So the amount of energy we're putting into this is enormous. To produce those, the fuels, the chemicals, the plastics, all of the smart materials that we require requires a lot of energy, and that comes with a lot of energy production, and that's going to require a lot of carbon as much as we want to be shifting away from those sort of high-intensity carbon-producing energy sources. We still haven't quite got there, and then even getting there is going to be on the difficult side as well. But then actually on the material side of things is actually a huge problem in that the vast majority of materials, particularly plastics, are derived from oil-based sort of components, and sort of the petrochemicals that we have, we need to sort of use for these materials is just incredibly high, and so we have a very high carbon footprint just on the manufacturing of everything we need to have. And so the first problem then is all of the energy and the feedstock we have for making everything we need for modern life is just very carbon intensive. But not only are we making a lot of stuff from some very sort of pretty nasty feedstocks, we're not really getting rid of it either very well. And so we're generating a lot of rubbish, a lot of waste in doing this sort of, in our modern world. And as I'm sure we're all fairly familiar with, is that the waste we're producing is accumulating in a lot of our environment. And so we need sort of new methodologies really to tackle how we want to live in a modern world. And I'm not saying we should get rid of plastics or otherwise. I think that they are fantastic materials, and especially along with the other things that we've developed especially over the past 50 years, it's just, our existence depends on them. And so we need to, if we want to be able to kind of move forward and also sustain our planet in a way that's viable, is we need to both deal with the waste that we've generated, and we also need to shift the feedstock away from oil to more bio-based solutions and more sort of planet-friendly solutions. And so, for me, those two things can be done together. We can think of waste as a resource now, and so think, how can we best take everything that we've produced, the waste that we've produced, and how can we remediate that? How can we bring that back to things that then become useful? That waste to products is something that we're really interested in. And really, for my money, we don't need to look much further than biology. Biology is an incredible recycler. If biology wasn't reusing all of the chemicals and materials that it requires for developing everything that's part of us, part of the biosphere, I don't know how many times you'll be going to the toilet, but it will certainly be a lot. So, our biology is very good at reusing those materials. And so this is something that we want to harness. And for me, we want to use enzymes because they are nature's toolkit. So these are biomacromolecules that can perform a wide range of reactions. They can do pretty much everything conceivable that we would need in chemistry, but also materials production. And it does it really efficiently. They're highly effective sort of catalysts and sort of producers and reactants in producing the various things that biology needs. And so this comes with a very low energy requirement. Again, it's a very efficient process. These effective toolkit and this ability to perform loads of reactions, and all the reactions actually we could possibly have conceived of as chemists, but was very efficient. Unfortunately, though, we've developed a series of industries and production methodologies which are slightly incompatible with biology because they're not very stable. For some very good reasons, enzymes are very minimally stable, so they don't tolerate too many sort of changes in temperature, and the window of usability is very, very low, and this can pose problems. But not only that, is they only really work in water, so water is the solvent of life. It is where biology has evolved to be so efficient, but that's not what we're using as a in our modern world. We're using solvents which are sort of non-aqueous in nature, so that they're not water, they are completely different. And so what I'm really interested in doing is how can we bring biology and drag it out of its comfort zone and harness it for our purposes, because really it's done everything we need to do, we just need to harness it somehow. And so my research is on developing a modification strategy where we chemically protect our enzymes. We can install this coat around the enzymes which will do two things which are very key for us, and that is, it actually improves their stability. And I'll show you, going to maybe trigger warning, there's going to be some science. I'm going to throw some science at you, and just, I can't help myself, that's what we do. So they're becoming very stable, but also we can use this protection methodology to drag them out of that solvent for life, water, and we can get them to work in different solvents. We're doing a lot of different work on sort of how we can explore the use of enzymes that we've done this chemical protection to, but the one we're going to focus on today is plastics, and actually particularly the plastics that are used in sort of those single-use plastics. And actually, I'm going to focus particularly on a type of plastic which is nominally called a bioplastic, and also that is often labeled as being biodegradable, often labeled as being not home compostable, compostable. People think that's home compostable, but it's not. It's these plastics are sort of usually sold or distributed as being bio-friendly. I guess the key operative there being is they're just a little bit more friendly than other plastics. And the plastics that they often appear in is in this kind of cup. So for often you find them in beer gardens. Actually, I've noticed recently that some places have shifted back to oil-based plastics, which is, I don't know, 6 of one, half a dozen of the other, I guess. But often those coffee cups and the plastics used in sort of this disposable packaging is obviously quite common. This plastic is polylactic acid, or PLA. You might notice it if you look at the recycling codes. It has a recycling code typically of 7. And this is something that most authorities in the UK cannot deal with. There is only a handful of sort of routes to recover this material and recycle it, and the vast majority of sort of local authorities don't have the capability. And so we want to be able to see how we can better use this material and better recycle the material so it becomes circular. And looking at the synthesis strategy then currently because it is bio-based, you can make this from crops. So from corn, you can extract the lactic acid. You can polymerize this to the polylactic acid, which you can then make to form your cups, etc. The most common route to then get rid of this is to go to either the rubbish or it's incinerated. The few places that can recycle it will typically go through a process of industrial composting, which is a very slow process and is actually relatively energy intensive, all things considered. And you get a product which is, whilst useful, isn't necessarily that high value, and you can start seeing where problems might come out of it, in that we're just going to get compost out of this process. In theory, you can close this loop by growing your crops again in this compost, but if we start wanting to shift away from oil-based chemicals and oil-based materials, this is going to be in huge competition with our ability to make food. Agriculture is not going to be happy with us if we're just going to start shifting all of our land use to making bioplastics. And so we really need alternative strategies to try and make bioplastics, these materials which we use a lot, but to be able to reuse them again in a way that is much more efficient and allows us to build materials as if they were brand new again, which is the focus of the work we're doing. So here comes the science. So we've taken an enzyme. It's a fairly common enzyme. You might not know it, but you will know it because this is an enzyme that's found in laundry detergents. So any of those bio-based laundry detergents you have will contain quite a lot of lipase. This is a protein, an enzyme that breaks down fatty acids normally. And so I'm not going to explain too much, but just to kind of illustrate our point, so just, again, a bit of data, I can't resist, is that through some very unique experiments we've performed, we've shown the window of stability for this enzyme after we've modified it is between 20 and 120 degrees Celsius. So we've really shifted that window of operation well beyond what is possible in both water and for biology normally. And I'm going to cut a very long story short and say that what we can do is we can take plastic cups. So these are snippets. This was a Hotel Chocolat cup, I believe, that students diligently cut into 100 milligram chunks. And they were able to show through a change in the solvent to something that's more amenable to plastic dissolution and protecting our enzyme so we can operate at higher temperatures, we can degrade this plastic, this post-consumer plastic within 24 hours. We can completely dissolve it. It's gone within 24 hours. And not only that is, within 2 days, and within 40 hours, we've converted all of that plastic to its constituent building block, the monomer. And so this was really quite exciting because what we've effectively done here is going back to our loop here, is we've short-circuited it. We can go straight from the plastic back to the monomer, and now we've got a fully circular system for producing a plastic that is of equivalent quality to what was there before, but without the requirement for having to go through industrial processing or without having to use up all of that land to make the virgin materials. And it only happens in 2 days. And so this is really quite exciting stuff that we're starting to work on. We're not just focusing on plastics. Plastics is obviously where we're starting with this is just the start. We're going to be looking into textiles. We're looking at food to fuels, and not just the traditional biofuels, but fuels which we can use in all kinds of different processes, and a whole lot of other things as well. And actually, something that we've noticed, and we've published a couple of times on this now, but something I want to delve into more is actually this modification strategy isn't just good for making stuff and recycling things. It actually protects biologics from degradation. And so actually what we can do is we can drastically improve the shelf life of protein-based therapeutics. And so things like antibodies have a significantly greater lifetime, to the point where we can envision a world where we can deliver these therapeutics without the requirement for the cold chain, so without the requirement needing a fridge or a freezer. Really, this is a nice opportunity for us to start democratizing access to those medicines that we really benefit of in industrialized areas. And so with that, I just wanted to sort of conclude there, and I can't resist, and I have to thank everyone who's contributed to this, so my wonderful group, particularly Susanna, who led on the polylactic acid work, the various collaborators over the years, and funding, and thank you very much for all of your attention here as well.